Hi everyone, welcome back to Dento'ZEN. Today we are here with a new video where we are going to learn histopathology diagram of ameloblastoma. So before we begin, I want you to quickly subscribe to Dento'ZEN if you have not done that till now. Also hit on the bell icon so that you remain notified about new videos. Now the term ameloblastoma has come from ameloblast. Yes, we heard about ameloblast in first year in dental histology in tooth development. So there we have a structure which is called enamel organ which forms from overlying epithelium. So in this enamel organ we have tall columnar cells which are called ameloblast cells, and above these tall columnar cells we have star shaped cells as we can see here, which are connected to each other. So these are called stellate reticulum cells. Stellate is star shaped, reticulum they are forming network. Now coming back to tumor ameloblastoma. In this tumor we see tumor cells which look similar to these ameloblast cells and stellate reticulum cells of tooth development. But they are arranged in different patterns. So based on that, we have 6 histopath types. So in the first type of ameloblastoma, these tumor cells may be arranged in follicles, that is round structures as we are drawing here. So the peripheral cells of these follicles are similar to ameloblasts, that is they are tall columnar cells, and the cells in the center of these follicles are similar to stellate reticulum cells. So that is the follicular type, that is the tumor cells they are arranged in follicles, and in between the follicles we have connective tissue. If these tumor cells they are arranged in long strands or cords which are connected to each other as shown here, forming a network, then it is called plexiform type. Plexiform means network. So the same tumor cells are here, that is tall columnar ameloblast like cells and stellate reticulum like cells, but they are within the cords or the strands which are connected to each other. Strands की form में पढ़े हैं, and in between the strands we have connective tissue, and within the connective tissue sometimes we can see cystic degeneration, एक space दिख सकता है. Coming to third type, acanthomatous type. Now it looks similar to follicular type, that is tumor cells they are arranged in follicles, and the peripheral cells of these follicles are similar to ameloblast cells, that is they are tall columnar, but there is a difference in the central cells. So the central stellate reticulum cells here can show a change, that is they can get converted into squamous cells. So there is squamous metaplasia of the central cells. Metaplasia means change. So that is called acanthomatous type, where there is squamous metaplasia of the central cells, and these squamous cells can produce keratin. Coming to next type, that is granular cell type. Again it looks similar to follicular ameloblastoma, that is the tumor cells are arranged in follicles, and the peripheral cells of these follicles are tall columnar ameloblast like cells, but the central cells will show change, that is they will have granules within them, and their cytoplasm is pink staining, that is eosinophilic pink staining cytoplasm, that is called granular cell. Now next type is basal cell type. Again in this the tumor cells are arranged in nests, but within the center of these nests we can see a different type of cells that look similar to the basal cells of the oral epithelium. So they are called basaloid cells, that is small cells with hyperchromatic dark staining nuclei. So that is called the basal cell type. Now coming to the last type, that is desmoplastic, which is quite different from all the types. Desmo means connective tissue, plasia means formation. There is formation of lot of collagen fibers as we are drawing here. These collagen fibers, that is dense connective tissue, will compress the tumor islands. So the tumor islands will get compressed. That is the main point. So the tumor islands like this, that is made up of odontogenic epithelial cells, they will get compressed. So compressed tumor islands, छोटे islands दिखेंगे, and the dense connective tissue is seen. So that is desmoplastic type. So those are the 6 histopath types of ameloblastoma. Now we'll see how we are going to draw each type. So in the first type, follicular type, we have to first draw the follicles, that is round structures, and within these round structures we have to draw 2 cells, that is peripheral cells and central cells. So first we'll draw round structures, peripheral cells, that is tall columnar cells, the central cells, star-shaped cells connected to each other. Now the peripheral cells will show the nuclear features, 3 important features. The first important feature is that the nuclei of all these peripheral cells are at the same level as we are drawing here. So that is called the palisading arrangement of nuclei. Second important point of nuclei is that all these nuclei are dark staining, which is called hyperchromatic nuclei. Hyper is more, chroma means color, dark staining nuclei. Third important point is all the nuclei are arranged away from the basement membrane of the cells. So that is called reverse polarity of nuclei. Now I'll put cytoplasm. In between the follicles we have connective tissue which contains blood vessels as we are drawing here, collagen fibers as we are drawing here, and the fibroblast cells, which are depicted here by blue color. So that is the connective tissue here. So we have follicles of tumor epithelial cells surrounded by the connective tissue. Let's do the labeling. So in the labels, first we have the ameloblastomatous follicles, that is, tumor cells are arranged in follicles. The peripheral cells of these follicles are tall columnar ameloblast-like cells with all the three nuclear features. Central cells are stellate reticulum-like cells, star-shaped cells, connective tissue stroma in between the follicles, and within some follicles we see cyst formation. So that is called intrafollicular cyst formation. Intrafollicular cyst formation. So that is follicular ameloblastoma. Next we come to the plexiform type. In the plexiform type, we have to first draw the strands or the cords of the tumor cells which are interconnected. Then within those strands, we have to draw the two types of cells, that is ameloblast-like cells and stellate reticulum-like cells. So first we'll draw the strands, then we'll draw very tall columnar ameloblast-like cells with all the three nuclear features, that is palisading arrangement of nuclei, hyperchromatic nuclei, and reverse polarity of the nuclei. These in between these cells we have star-shaped cells as we are drawing here, stellate reticulum-like cells, and in between the strands we have connective tissue. Within the connective tissue we have blood vessels as we are drawing here, we have collagen fibers, and we have fibroblast cells. At a place in the connective tissue we can show a space that will show the cystic degeneration, and will also fill the pink color in the cytoplasm of all the cells as shown here. So we'll fill the cytoplasm with the pink color. So let's do the labeling. So we have odontogenic epithelial cells, but they are arranged in anastomosing cords or strands forming network. And the ameloblast-like tall columnar cells are seen in the strands, stellate reticulum-like star-shaped cells are seen in the strands. Then in between the strands we see connective tissue, and we see cystic degeneration in the stroma, that is, in the connective tissue we can see cyst degeneration. So that is plexiform type. Next, coming to the acanthomatous type. In acanthomatous type, first we have to draw similar to follicular type, that is follicles with peripheral cells and central cells. Peripheral cells tall columnar showing nuclear features, central cells star-shaped cells interconnected to each other, connective tissue showing blood vessels, collagen fibers, fibroblasts. Now coming to the central cells, these central cells will get converted into squamous cells as we are drawing here, and they can show keratin formation. So these are the squamous cells as we can see here, and they can produce keratin. So it can be seen in the center of the follicle. So that is acanthomatous type and how it is different from follicular type. Labeling, so we have ameloblastomatous follicles. Peripheral cells are tall columnar ameloblast-like cells with all the three nuclear features you can label here. Then the connective tissue stroma is there, and central stellate reticulum-like cells are there, peripheral cells, central cells, connective tissue stroma in between the follicles, and squamous metaplasia of central cells of the follicles. That is the important points. That is, the central cells of the follicles, star-shaped cells, will change into squamous cells as we can see here. So these star-shaped cells, they are changing into squamous cells, and there is keratin formation. So you can label keratin also here. So that is acanthomatous ameloblastoma. Next we come to the granular cell ameloblastoma. So again in this, first we have to draw similar to follicular type, that is follicles with peripheral tall columnar ameloblast-like cells and the central cells and the connective tissue stroma in between the follicles. Now we'll show the change in the central cells. So now they are granular cells with the granular cytoplasm and eosinophilic cytoplasm. So as we can see here, these are large cells, they have granules in them, and their cytoplasm is pink staining, that is eosinophilic. Now we'll do the labeling. So we have ameloblastomatous follicles, and the peripheral cells are again tall columnar ameloblast-like cells with all the three nuclear features. The central cells will show change. In between the follicles, we have connective tissue stroma. Now, the change in central cells is that they will show granular cell change, and these granular cells have eosinophilic cytoplasm. So, those are the two important points that you have to label here. That is granular cell change in the central cells, and these granular cells have eosinophilic cytoplasm. Next, we come to the basal cell type. So, again, we have to draw it like follicular type, but islands, some tumor follicles will show the peripheral cells which are cuboidal, and in between them we have connective tissue. So we are drawing the cuboidal peripheral cells, and the central cells of these follicles are basaloid cells as we are drawing here. So they are small cells with dark staining hyperchromatic nuclei in the center. So that is basaloid type cells. So this is called basal cell ameloblastoma. So let's do the labeling. So again we have ameloblastomatous follicles which have peripheral cells and central cells, and in between them, in between the follicles, we have connective tissue. Now here the peripheral cells are cuboidal shaped cells, and central cells are basaloid cells. So that is important point, that is basal cell like cells, that is basal cell ameloblastoma. Next, coming to desmoplastic, it is different from all other types. So here the tumor islands are compressed, so we'll draw small islands and compressed islands. So we'll draw small tumor cell island with connective tissue, and then we'll draw a compressed tumor island as we are drawing here, where we cannot distinguish between the two cell types. हमें नहीं पता चल रहा ameloblast cells and stellate reticulum like cells. Then another island, compressed island, and dense connective tissue showing lot of collagen fibers as we are drawing here. Another compressed tumor island and the connective tissue. So that is desmoplastic type. Now the important labels here are, that is compressed tumor cell island, as we have drawn here. So compressed odontogenic epithelial cell island, sometimes it gives kite tail or kite like appearance, and the connective tissue is desmoplastic, that is desmoplastic stroma, that is there is dense connective tissue. So that is all for this video. I hope you enjoyed the video. Useful tip, you can use this video to attempt short note on histopathology of ameloblastoma in your theory exam. So if you really liked the video, tap on the like button. Also share the video with your friends. Keep watching, keep learning. See you in the next video. Till then, take care. Bye-bye.